Thank you for giving me the time to present. So, Harae Dx is a 3-year-old company. It was started in 2021. Um, it's meant to develop diagnostic products where we are starting off with cancer diagnostics and specifically for initially the test is for breast cancer. Um, the company it's we developed it so that it can be used in rural settings. Today, cancer diagnostics is available only to people in urban sectors. So less than 2% of the world's population has access to it, especially when we talk about breast cancer. As an example, which we are developing, there's over 3 billion, 1.6 billion women over the age of 40 who need to be screened and tested for breast cancer. However, today, there's only 60 million mammograms which are conducted. And so the number of the amount of cases being tested is only less than 4%. And it's a big problem. And most of the population worldwide, about 60% lives in rural segments or semi-urban segments, and they don't have access to it. So cancer is generally found at a very late stage. And that's, when you take breast cancer, for example, or cervical or ovarian cancer, you find it at a stage when it is at stage 3 or stage 4, unless you have access to mammogram, which most people don't. So, and the test which is available is mammography or ultrasound. The problem is most women do not want to go and get these tests done, basically because they're invasive and very painful. So we have developed the first portable cancer diagnostic technology, and it's not just for cancer diagnostic, it can be used for anything, but initially we are targeting cancer. And so if you look out here, it's a microfluidics technology where you have a disc and you can test 6 patients on this one disc. There's an instrumentation similar to what you do with COVID testing. You had the strip and you would put a sample of saliva or nasal. In this, you take a couple of drops of blood and you actually put it into the disc. And then you've got these antibodies placed in the white area that you're looking at, the white spots. There are 5 different biomarkers which we are testing for. And then the AI looks at that and combines it. And these markers, if you look out here, uh, it's not working. But if you look out there, these 5 markers are generally separated out quite clearly. Uh, the pink is where they're in the normal zone, and the orange or red is where they're in the cancer zones. Okay. So, unfortunately, so using AI, we are able to take all these together, and the system can learn and pick which are these, if someone's got cancer or no, depending on which markers are upregulated and which are downregulated. This technology was initially developed at Lawrence Livermore National Labs. However, it was based on an array system, and we moved it to to a biomarker testing system using ELISA testing or sandwich ELISAs. Since then, we tested about 120 samples in our first study, and then another 92 samples in our second study, just to prove that the biomarkers work by themselves. And you can see out here, we're getting over 91% sensitivity and specificity. Now, you compare it to mammograms, which is the standard of care, that's less than 70%. So, and most Most people don't want to get mammograms done because it's painful and it's expensive. It costs over $200 to $300. This test that we are developing is going to cost less than $10. Okay. And it's a simple draw of a blood. It takes 15 minutes to get the test results done. Subsequently, we tested this on the earlier prototype instrument that we developed, and we did 7 other studies, going from the smallest study with 56 samples, and then the largest being about 308 samples. And we got a sensitivity and specificity of over 85%. Now, the system needs to be trained further to get it back to the 91%. we have showcased it to physicians throughout the world, in India as well as the US. Dr. Ralph Smathers, who was head of mammography at Stanford, left and started his own mammography practice, uh, and others in India and other places. And we also showcased it to the Indian government, and we've spoken to the US FDA. Dr. Reena Phillips, who's head of IVD at FDA, has said that this could qualify for a breakthrough and a fast track, which means in about 9 months we could have approval once we go in and submit documentation. The Indian FDA, which is called DCGI or CDSCO, has already done a clinical registry on this, waiting for us to finish up the product. And subsequently, they have given us, in the state of Gujarat, they proposed a testing, and in the state of Maharashtra, they proposed testing using this as a pilot test, to screen 10 million women. In Lucio County in Sichuan province, they have asked for bringing the product there to do a pilot study of 1.6 million women using this technology as well. Uh, the model that we have to market this, there are 2 models. The initial model, there are 2 models. One is to make the test available using a physician-operated lab, and we are starting with that model in Thailand. We've got a group there where, as the funding comes in, we'll start off with that, and it also allows us to get more data to train the algorithm. The second part of it, I shouldn't say 2 models, but that's the first part of it. The second part of it is to bring the instrumentation, which will cost about $2,000 to a physician, and the kits themselves will cost about, we look at selling it about $60, and that's for 6 patients. So it costs about $10 a patient. And if the physician decides to mark it up, they do it for 20, charge the patient $20. That's about the same price as you would do a copay, and that's much less than mammography. Now it makes it available to a lot more people. As the volume pick up, this is expected to go down even further, and we expect to bring it down to about $10, between $5 to $10 a patient. Now, who is in this field? There are companies like Illumina, which bought Grail. Illumina has been doing it based on genomic testing. Roche, with Foundation, has done tissue sample, Exact Sciences, Guardant Health, Freenome, etc. But none of them have a portable system which can be taken into rural communities, which is what we have. And nobody can do it at a cost point that we are talking about. So while these are competitors, I believe that in the long run, they would be companies who would acquire Harae Dx. Our initial area that we're looking at is Mammo Alert, which is for breast cancer. Then we expect to go into pan cancer, lung cancer, thyroid, ovarian, and cervical cancer. Our focus is mostly initially around women's health in regards to cancer. Management team, everyone is working part-time right now until we get our funding in place. Uh, I'm uh, I'm a serial entrepreneur. I started this company. I started several companies, and then we've got other teams, other members in the team who are working as part-time until we get funding, and then some of them will come on full-time. Others will continue with their other ventures. Um, I myself had run a company called POC Medical Systems, where we raised 30 million before this, and prior to that with Actus Biologics, where I'd raised about 15 million for cancer therapeutics in India. And uh, with my father, started the first breast cancer diagnostic company as far as computer-aided diagnostics is concerned, which we sold to R2 Technologies, which was bought by Hologics, and that's the standard of care throughout the world today. The technology was developed by us. Where are we? We are looking right now for a bridge round of 3 million. We have actually signed a term sheet. Well, we have signed a share purchase agreement with ultra high net worth individual out of our group of individuals out of India for 15 million. Uh, that's going through the Indian RBI approval process before the money can be sent to us, which will take us another 3 to 6 months to get that. So in the meantime, we're looking for about 3 million to keep moving forward. Also, we have had funding. This technology, by the way, the data which I showed you was developed at POC Medical Systems, which was my earlier company. When COVID hit, we had to shut down the company because all the funding was going towards COVID and not into this area. BioVentures, Fusion Fund had invested in that company at that time. Sun Walking Angels and SOSV has since invested in the company. So as I said, I exit strategy, key selling points. We've got 5 provisional patents, 5 patents which we've got licensed from the acquisition of the assets from PLC. And we have another 3 patents we are expecting to file. Uh, we, when you look at the model, there's revenue coming in from these methods, but also there's the AI part of it, the data which is accumulated is something the companies like Pfizer and Genentech and others who are working in cancer therapeutics would like to get that because today they have less than 5% of the data. The world's data doesn't exist with anyone. There isn't a device like this available today. Getting a device like that will make data more available to these people and they want the data. So that's a separate revenue stream, which we have not even talked about. Potential exits could be some of them buying it, but we believe it would be a Roche, Exact Sciences, Guardant, Logix, Illumina, or AbbVie who want to access this area. So in summary, we've got a technology which is focused around checking for protein biomarkers as opposed to genetic biomarkers, as you saw. And the reason we picked protein biomarkers is protein biomarkers tells you what is happening today. Genetic biomarkers tell you what can happen in your life. It doesn't mean what is happening today. So that's why we have picked protein markers. We have data which shows it works. We've also have a pipeline which we believe is going to be of major use, and there's a market for that. When you look at global markets, uh, when you look at the Middle East, you look at Asia and some of the other countries where the largest population of women live, they do not want to go and get tested using other technologies where they have to disrobe. So as a result, not just because it's not available to them in rural communities and even in urban communities, it's also because of religious as well as other issues, social issues that they don't want to get tested. With a simple blood test, which takes about 15 minutes, they would want to get tested. And when you look at urban sectors, women today are very busy with their jobs, families, etc. And if they have to go in and schedule and get a test done, it takes too long. Here they walk in while they're waiting for to see their doctor, the test is done, and in 15 minutes they have the results. With that, they have the app where they get this data, they can follow their breast health score. So there's a Mammo Alert score, which is just based on the protein markers, and then there's the breast health score, which is based on uh, on their history as well as their family history, etc., and their age, etc. So that's what we've got. So that's about all I have. If there's any questions, I'll be more than happy to answer.
It looks like you use the antibody or just like flu, yeah. The antibody to capture your biomarker, yeah.
Correct.
For breast cancer, you capture 5 different biomarkers
Correct.
at the same time.
Correct.
So, and you have a disk actually, the centrifuge, then they distribute the serum. And so what method do you use to visualize your band?
So it's a sandwich ELISA, and if we go back here,
like ELISA, yeah.
Yeah, it's a sandwich ELISA.
Yeah. So what are you labeling on that? Is it use enzyme or use fluorescence or?
We're looking at fluorescence.
You look for fluorescence.
Correct.
Okay. Yes. So
and so one of the issues with fluorescence is you're not able to go down to pico levels. With what we have developed, we can actually go to pico levels as well.
Yeah, fluorescence is some trickies on labeling, so that the numbers is very variable. So, but the other questions you use, like for example, 5 different markers.
Correct.
So each marker they have different expression levels.
Correct.
Especially on the breast cancer.
Correct.
How you evaluate which one it can be, like for example, and above the threshold, for example, you have to determine that. Yeah.
So when you go back and look at it's not looking at so when you look at these, each of them has got different thresholds, right? But we are not looking at one biomarker. We are looking at a combination of biomarkers. Yeah. And so it's not that this has to be above the cancer level. What we have found is even if 2 of these biomarkers are in combination are at a higher level, that actually means that they have cancer. So it doesn't have to be that each of these biomarkers is past the level. So we are finding a lot of things which was not known to the whole market before. So it could be what you find is if 3 markers are up regulated, that person's got cancer. If it's 2 markers are down regulated, they probably don't have cancer, but they have to be watched for that they can develop cancer in their lifetime, right? And so now instead of doing a test every 2 years, you have them come back and do it in 6 months or a year to watch if those biomarker levels are changing.
So you use that machine to rate and to centrifuge?
It does the whole thing. Yeah. So you just take the blood and you put it in these spots that you're looking at in the middle. And the blood, as it spins, the plasma and the blood cells separate out. And then the plasma travels down into the area where actually it travels down. Let me point it out. It's not going. It travels down into the area where the antibodies are placed, the white zone. And then as it travels through that, each of the protein markers is absorbed on the antibody, and the antigen also comes along and it fluoresces, right? The antigen's got the fluorescent tag, so it fluoresces, and we look at the intensity of the fluorescence. So we are not looking at what is the absolute, well, the intensity tells us the absolute value.
Okay, you can quantify, you can get the,
correct,
quantity of the each
Correct.
Each density, yeah, each
Yes.
marker. So you said one disk you can test 5 patients. That's
6 patients.
6 patients. Okay, you have 6 well.
Correct.
So each well you have multiple channels.
Yes.
Okay, all right.
So in the long run, when I said we are looking at a pan-cancer disk, you don't need, in that case, it will be one patient per disk.
Yeah.
And instead of having 6 times 530, you can actually have up to 60 antibodies placed in there.
Okay.
And then it can look at all those and tell you which is up.
So you manufacture the disk by your own?
No, we have it contract manufactured.
Okay, so your disk open to the other developer, for example?
We have right now we have not made it available to anyone. Right now we are
just for yourself right now.
Correct.
But the machine, yeah.
Later on, as we go along, we make it available to the whole biotech industry. Our focus right now is
for your own, yeah.
Exactly.
Okay,
this technology doesn't exist, so people are not able to use that, right? I mean, it doesn't exist to everyone else. It's
okay, all right.
Proprietary.
Yeah. Okay, thank you.
Okay.
Yeah.
This are you already testing patients now, or it's just prototype?
We don't need patients to test this because patient samples are already available in different hospitals and blood banks, and we can get the plasma samples and test it to show that retrospective. We don't have to do. So we have spoken to FDA and others. We don't need to get new patients to test it. The comparison is against mammography, which is the standard of care. So we take samples which have gone through mammography and biopsy, and against that we compare it.
Business-wise, it's not started.
No, not yet. We did a lot of the work, as I said, at POC, and unfortunately, during COVID, we were raising our Series B. We had to shut it down because nobody was at that time. All the money was going there, so we restarted the company. We got some money in. We have signed the agreement to get the rest of the 15 million. And once we finish this, because it was an earlier version of this disk and an earlier version of the instrument. So those were prototypes. This is the design for manufacturing that we are working on. And so we have optimized this further. There we were using beads. Here we are using antibodies put on a membrane so that with beads and other things, you get issues where beads get stuck in the microchannels. In here, you don't get all those issues. So we have optimized it to be able to do it in large scale.
So you use beads?
Earlier, we were using it, no longer. No longer. Beads get stuck in the microchannels. So then you don't get signal.
So you coating your disk by your own, or
yes.
You use outsource?
Right now, we are outsourcing everything. Everything because with a small company, we don't have the everything, the different components and stuff like that. It costs too much to set up the whole infrastructure. So we are outsourcing each of these components. It's not just one thing or the other, right? Yeah. There's the software component, there's the instrumentation component, there's the microfluidics component, and then there's the membrane with the antibodies components, right? Each one of them is separately outsourced, and then we assemble that.
How big is the device? It's a portable, so how big?
It's this big.
Oh,
literally. And if you look up there, that's a little handle, you can actually carry it. The screen is a 5-inch screen, and all the data is displayed there. And if you have your app, the patient can also get it. The physician has a physician portal where they can go in and look at all the patients. You would be able to look at only your data, and we can look at all the data.
And then you say it takes like 15 minutes to get the result. And how many kind of diseases it can take at
So right now, our initial work that we are doing is with breast cancer.
only.
But we plan to go into ovarian and cervical cancer, and then go into also pan cancer, so lung cancer, etc. But it's basically a sandwich ELISA machine. It's an immunoassay machine, nothing else, right? Yeah. So
Anybody you get, yeah.
exactly. And so what we are doing is through one of our partners, which is my sister's company, we are doing the recombinant antibodies ourselves. So we are not off-the-shelf antibodies are not as as good. They vary. So we are doing our own.
We can provide all kinds of antibodies.
Yeah, so we can talk about that. Yeah.
How many blood do you need?
You need a couple of drops, just 2 drops. So you remember, this is actually, we have already shown it. We have done this work, and we've got 15 people in the company, all working part-time right now because we don't have funds to pay everyone's salary. Everyone's got equity, and so additional design work is going on. So, but it takes just about 2 drops of blood.
It has to be liquid. Dry blood doesn't work.
No, it's got to be liquid. So it's fresh blood or plasma. You can take plasma and do it.
Okay,
with that
thank
as
you.
well.
Okay, thanks.